So good morning, students. Good morning, students.
Good morning, sir.
Good morning, sir.
Morning, sir.
ओके तो इसका मतलब सब जाग चुके हैं, ठीक है? ओके, सो एनी क्वेश्चंस? अभी तक हमने जितना पढ़ा है, वी हैव talked about history of immunology, we have talked about antigens, we now know a lot about antibodies as well. So the way I am teaching, the way I am explaining, is it clear to everyone? किसी को कोई issue समझने में या any problem that you are facing?
Yes, sir.
Okay. Okay, so students, if you remember last time in the last class, we talked about the hinge region and we also talked about IgG, right? So just raise your hand to tell me which of the antibody classes do not have the hinge region. Okay, my question is which antibody classes do not have? Okay, Nehal.
Sir, IgE and IgM.
Okay, good. So IgE and IgM does not have the hinge region. Rest, other three types, they have the hinge region. Okay. My another question is, so Nehal, you can remove your hand. Okay. Another question is, when we do the sequencing of hinge region, so it is rich in which amino acids? Yes, Manaswani.
So it's proline and cysteine.
Yes, fine. So proline and cysteine. So my next question is, what is the importance of proline? What is the importance of proline in antibody structure? Okay, okay. Let's try Tanvi.
So, it prevents folding in a globular structure.
Yes. So proline, it prevents the antibody to get into a globular shape or structure. Okay, and therefore it helps in maintaining the Y shape of the antibody. And what is the importance of cysteine? Okay, so Shakti.
Yes, sir, it is involved in the formation of disulfide bonds, interchain disulfide bonds.
Yes, so it is important for the formation of interchain disulfide bond, right? And because of those bonds, the different chains, the heavy chains and the light chains, they are held together. Okay, so if we just break those bonds, like we can use mercaptoethanol and break those bonds, and we can separate the chains, right? So cysteine is very important in the formation of disulfide bonds, and therefore it is rich in the hinge region where actually it is holding the two heavy chains together. Okay. Okay, so that was about hinge region, and then we talked about IgG, right? So I think you remember that this is the most abundant immunoglobulin present in us. okay, and 80% of the total serum immunoglobulin which is present in our blood, IgG is 80%. So just imagine 80%, or इसके अलावा there are four more classes, right? So IgG itself is 80%, तो बाकी की जो चार हैं वो उस 20% में fall करेंगे, okay? So how many subclasses of IgG? Yes, Devanshi?
Four.
Okay, so do you know the difference between the four? Anyone?
Sir, can you repeat the question?
What is the major difference between the four subclasses of IgG? Okay, Manaswini, go ahead.
Sir, so one of the differences is the number of disulfide bonds in each. So
Mm-hmm.
IG1 has 2, IG2 has 4, IG3 has 11, and
Mm-hmm.
IG4 has 4.
Yes, fine, fine. Okay. Okay, students. So it shows that uh, things are clear to you. So last time we talked about hinge region, IgG. So most of the points are clear. Okay, and so last time we also talked about FC receptors. We talked about phagocytic cells. We talked about opsonization. Right. So I hope all these things are clear to you. So let me now share the slides and start further. Okay, just to revise IgG, uh, I already told you that it's the one of the most abundant immunoglobulin present in us. Okay, so almost 80% of IgG is present. There are four types: IgG 1, 2, 3, and 4. The difference is the size of hinge region and the number and position of interchain disulfide bonds. Okay, so this is the picture. So Manaswini already numbered them. So we have two disulfide bond, we have four disulfide bond. I think we have 11 here and four in IgG4. So this we also discussed. So each type has a different role, right? So again, students, if nature has created four different types, then it must have given different roles to those different types, right? So obviously these different types have different activities. Some we know and some we also don't know, right? Okay, this again we have discussed. So now we are going to start with IgM. Okay. Now again, this is also very important immunoglobulin, and it's 5 to 10% of the serum immunoglobulin, right? So out of 100, right, 80 is IgG, and then 5 to 10 is IgM. Okay, and IgM is also expressed as membrane bound antibodies. Okay, ये मैं एक बार अभी क्लियर करता हूँ. So see what happens. अभी तक हमारे दिमाग में एक इमेज है एंटीबॉडीज की, that they are always secreted and they are always circulating, right? This is the image we have of of antibodies, right? So that is true, but some antibodies they are also membrane bound. Okay, just for let me draw it here. So suppose this is your cell. So IgM and IgD, they also express as membrane bound, right? So they are bound to the membrane, and so they will be like this. So they're membrane bound already. Okay. हमने जो कल बात की थी वो था कि सर्कुलेशन में हैं and they bind to the receptors through the FC region, right? But some of them are expressed as membrane bound, so they are already there on the membrane. Okay, so they are there with their antigen binding site on the outside, and if some antigen comes, it can bind over here. Okay, so on some of the B cells, they are also expressed as membrane bound. Okay, और इसको लिखने के लिए we just put a small m before IgM. Okay, so if we are putting this small m, it shows that it is membrane bound immunoglobulin M. Clear? Now the most important thing of IgM is that it's the first immunoglobulin of primary immune response. Okay, so primary immune response is the one which gets initiated when the immune system sees an antigen for the first time, right? So it's the first immunoglobulin of the primary immune response. Okay, and it is the first to be synthesized by neonates. Okay, so IgM में बहुत कुछ first है, so it's the first one, first immunoglobulin to be synthesized by neonates, and in adults later on in the life, it's the first immunoglobulin of primary immune response, right? So I don't know, अभी recently जब COVID हुआ था, so again we were talking of IgM, IgG antibodies, right? Even for dengue we say कि IgM, IgG antibody test करा के लाओ, right? So I will come on to the significance of that in I think uh, after 2-3 slides. Okay. Now, another very important thing of IgM is that it has a high valency, right? So theoretically, it can bind 10 antigens, right? So you remember the structure of a pentamer, right? So there were 5 antibodies held together and the antigen binding site was outside, right? So it has a high valency, it can bind to almost 10 antigens, right? But even then it does not, right? But it can still bind up to 5 antigens, right? Due to hindrance, right? So sometimes what happens, the antigen may be big, so it will hinder and it will not allow each site to bind to the antigen, right? But still you can imagine that it can bind up to 5 antigens, right? And it is more efficient than IgG in complement activation, right? So it's one of the best activators of the complement system. Okay, so again we will talk about complement system in detail after we complete antibodies, right? So again, students, you can just see so many important things here. 5 to 10% in our system. It can be membrane bound also. It's the first immunoglobulin of the primary immune response. Okay, we are not saying secondary immune response, right? So first immunoglobulin of primary immune response. It's the first one to be synthesized by the neonates. It has a high valency. It can bind to a large number of antigens, right? And it is most efficient in activation of the complement system. Okay, clear? And students, again, uh, why there is a need for a pentamer, right? Just imagine कहीं किसी की बॉडी में कट लगा और बहुत सारे एंटीजन अंदर घुस गए, okay? बहुत सारे पैथोजन have entered, right? Now सबसे पहले कौन जाएगा? IgM, because it's the first immunoglobulin of primary immune response, right? So now just imagine if you have to fight many antigens, right? मैं एग्जांपल देने वाला था कुछ और. So suppose एक घर में बहुत सारे चोर घुस आए, right? So you need a bigger army, right? One person cannot go and fight, right? So you need a big army. You need a lot of individuals to fight, right? So those individuals, they can group together and then they can go and fight and eliminate the thieves, right? Whatever you can say. So in this case also, when there is a cut in the body, so a lot of pathogens will enter, okay? So the number would be high. So you need a big army. So a pentamer is formed. The pentamer goes there and it tries to bind to as many pathogens or as many antigens it can bind. Okay? So that the number can be reduced. Okay? Clear? Now just imagine hundreds and thousands of antigens have come in and a single IgM is going and trying to bind, right? And just imagine if a pentamer is going, it will be binding more and it would be more effective in controlling the numbers, right, in less time as compared to if single IgM molecules are going there, right? So that is the importance of having a pentamer. Clear? Okay, so you can see the picture of a pentamer here. So as I already showed you, the variable region is on the outside. Okay, and this is the point where they can bind the antigen, right? So it binds through the CDRs. So this you know, right? And this is, so I'm just going to tell you, again I will not go into details of this, but just to give you an idea how a pentamer is formed. Okay, so pentameric version is secreted. So we know that 5 monomer units, they're held together by disulfide bond. So you can see there are 5 IgM molecules which are held together by these disulfide bonds, right? And where are these bonds formed? They're formed at CH3 CH4 domain, okay? So bonding between the molecules is at the CH4 and the CH3 domain, okay? FC region is in center, so you can see the FC region is in the center and the antigen binding sites are on the outside. Okay, so pentamer contains additional FC linked peptide. So you can see when I'm saying FC linked, it means it's linked to the FC region, right? So you can see this is the FC region and it has an additional polypeptide chain which is called the J chain. Okay, and J, you can say it's the J or the joining chain, right? So this chain helps in joining of the 5 monomers. Clear? So what is the role of J chain? It helps in joining of the monomers to form a pentamer. Okay? So, okay. So is this image of IgM pentamer clear? So you just need to remember few things. So 5 molecules, FC region is on the inside. These molecules are held together by disulfide bond at the CH3 and CH4 domain, and there is one extra additional J chain which has helped in the formation of this pentamer. Okay. Now let's see the steps how this IgM a pentamer is formed, right? So students, because of less space, I cannot show the full antibody structure here, but you can just imagine this is CH3, CH4, CH3, CH2, and here it would be CH1, and then your variable region, right? Similarly, it would be here, and then your light chain. Okay, so this is your antibody molecule. Okay, so I'm not showing the other two domains. Okay, okay, so what happens? First, you have one molecule of IgM, then another IgM also appears, right? So two IgM monomers are formed in the endoplasmic reticulum. Okay, and as I said, only the Fc region is shown. Now, cysteine in the J chain. So this is your J chain, right? This yellow one is your J chain. So, okay. So cysteines, and these are the cysteines in the J chain form disulfide bonds with cysteines from each monomer to form a dimer. Okay, so you can see these cysteine here in this J chain and the cysteine in the CH4 domain, right? They form this kind of bonding. Okay, so they form disulfide bond. So you can see the J chain is removed after disulfide bond is formed. Okay. Okay, so we have seen this point, right? So J chain came, it helped in the formation of disulfide bond and it gets released. ठीक है, ये disulfide bond बन गया, and this will now held these two molecules together, right? But we have to make a pentamer. So the J chain, it detaches, leaving the disulfide bond, okay? And then now it will again form a bond with the third molecule, right? So you can see J chain will now form the disulfide bond, okay? So similarly, this will keep occurring. Again, there is formation of disulfide bond, okay? The cycle is repeated twice to form this, okay? And then finally the J chain remain attached to the IgM pentamer, okay? So जो final bonding है that was through the J chain, right? So वो disulfide बनाता गया, okay, and finally it stayed here, okay? So at last point it is not detaching. So whenever you see a pentamer or when you see the dimer, okay, you will always see the J chain, okay? Now just remember students, you saw one of the pictures in previous class. Remember this? So you see here the J chain, right? So in a pentamer, there is a J chain. Similarly, in a dimer, right? So this J chain will basically help in formation of dimers, trimers, or tetramers or pentamers, right? Because नाम में ही है J or the joining chain, so it helps in joining. It helps in the formation of disulfide bond between the cysteine residues. Okay, clear? Any questions till this point? So you can see this animation again. So one molecule of IgM, right? Second molecule of IgM, J chain will help in formation of disulfide bond between these two molecules. Okay, so once the disulfide bond is formed, it gets detaches and then it attaches the next molecule, again helps in the formation of disulfide bond. Okay, again it helps in attaching the next molecule, again there is formation of disulfide bond, and then finally the fifth molecule, there is formation of disulfide bond, and at this point the J chain does not detach itself, and thus it leads to formation of the final pentameric form of IgM. Okay, so is this clear process? So it's a very simple process. The only thing is there are different steps, right? Nothing much. You can just remember that J chain is helping in the formation of disulfide bonds between the cysteine residues. That's it. Okay? Any questions?
So I have a question. So,
Yes.
after the formation of the pentamer, if let's say somehow the J chain detaches, then will the pentamer break apart?
Uh, See, it may not break apart, right? Because the other molecules are held by disulfide bond, right? But it may not form the proper pentameric form, okay? And uh, this pentameric form is important in the complement activation, right? So suppose if this happens that the J chain detaches, which will not, but suppose like if it detaches, then obviously you can imagine that one end will always be loose, so it will not form the proper pentameric form. It may be effective, right, in binding to the antigens, but it will not be effective in activation of the complement pathway. Okay.
Okay, sir. Thank you.
Okay. Any other question? Okay, so let's see the clinical significance of IgM. This again is very important. Okay, so as I said, IgM antibodies, they appear early in the course of an infection, right? So as I said, these are the first antibodies that are produced in response to primary infection, right? And usually reappear to a lesser extent after further exposure. Okay, so IgM antibodies will appear only for the first time. Okay, and they usually reappear to a lesser extent after further exposure, right? So suppose a person gets infected with dengue for the first time, right? So you will find IgM antibodies, right? But now suppose the person is uh, like after 4 years, 10 years, it again gets dengue, right? Now this time you will find IgG, right? And so this is another thing, right? Another point is like today a person got infected, right? So you will find IgM, okay, but maybe after a week you will find IgG, okay. So we'll come to this point again. This has a very important, it plays a very important role in diagnostics, this point. Okay, so we'll come on to this again. IgM antibody do not pass across the human placenta, so you can imagine we have already discussed it's only the IgG that can cross placenta, right? So these two biological properties of IgM make it useful in diagnosis of infectious diseases. Okay, so as I said, this property is very helpful, and we use this property in the diagnostics of infectious diseases, and I will discuss that. Okay, so demonstrating IgM antibody in a patient serum indicate recent infection, or in a neonate serum it indicates intrauterine infection. Okay, so let's just focus on the first point. Demonstrating IgM antibodies in a patient serum indicate recent infection. Okay, now suppose you go to the doctor, okay, and doctor thinks that you are having, suppose, dengue infection. Okay, so the doctor will recommend uh, dengue test, right? So now uh, you can either go for IgM or IgG, right? So if IgM is present, right, it means if IgM is positive for dengue, it means it's a recent infection, right? Because IgM is the first one to appear in an infection. Okay, but suppose you are not getting IgM and you are getting positive for IgG, it means it's not a recent infection. Uh, so either it is a bit old infection, or you may be having some other uh, issue if you already had dengue a few years back. Okay, so so if you just want to know that which is your recent infection, so then you should look for IgM antibodies. Okay, because IgG antibodies are also there as, so when the memory cells will produce antibodies, they will produce IgG, right? So is this point clear? So IgM antibodies in a patient serum, they will indicate recent infection. Okay, clear? Okay, let me show you a picture. Okay, let me just show you a picture to explain this. Okay, students, so you can see, so we are talking about antibody levels, right? So this is the incubation period, right? And at day 0, when antibody production starts, okay, so you can see early immune response, you will have IgM, okay, because as I said, the IgM level will be high initially, right? And then after 7 days, the IgG, or after 14 days, the IgG will pick up. Okay, so if you are looking for recent infection, right, when you are having fever, when you are having the symptoms, at those initial 7 days, you will find IgM in the blood, right? And later on, IgG will be more, right? So a combined IgG IgM test cannot distinguish between an individual's antibody response. Okay, so is this clear?
Yes.
Okay. Okay, so I think we can take a break, attendance break. And in the meantime, if any question appears in your mind, you can ask. And after attendance, we will start with IgA. Okay. So Adish,
Present, sir.
Adish, you are not visible. Okay, Atoiba.
Present, sir.
Am I visible?
Yes, Adish, you are visible now.
Yes.
Ayushi,
Present, sir.
Sam,
Present, sir.
Devanshi,
Present, sir.
Ishita,
Present, sir.
okay, Janvi,
Present, sir.
Kshitij,
Present, sir.
Manaswani,
Present, sir.
Namrita,
Present, sir. Present, sir.
Nandini, Nehal, Nehal,
Present, sir.
Nehal, you were not visible.
My
Rupal,
camera is on.
Sahiba,
Present, sir.
okay, Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridev, okay, Vivek, Vivek, Yukta,
Present, sir.
Yukta, you were not visible. Okay, Yuvraj, I can see you.
Present, sir.
Okay, okay. Okay, students. So let's start with the next antibody type, which is IgA. Okay, so IgA again, it is 10 to 15% of serum immunoglobulin, and it plays a critical role in mucosal immunity. Okay, so mm, So IgA is basically present in the external secretions of the body. So it is present in the mucus as well, right? So IgA is the most important immunoglobulin of mucosal immunity. Okay, so in our MSc program of MSc Immunology, we have a full course on mucosal immunity. So Dr. Pai takes this course of mucosal immunity, right? So this is a very important component of our immune system, and you can just imagine that this immunoglobulin, this IgA, it is present in external secretions, right? So IgA is present in milk, it is present in saliva, it is present in tears, right? It is present in the mucus of the bronchial, genitourinary, and digestive tracts, right? So it is providing protection, right? So suppose there are some pathogens in the mouth. So the saliva will have IgA, so it will tackle the pathogens in the mouth, right? Suppose some pathogen enters into your eyes. So again, IgA is there to attach to it, okay? And then the mucus of your bronchial tract, right? So you can understand like, so suppose a pathogen enters your nose, right? So it will go inside and then it will stuck in the mucus, okay? And in the mucus you have IgA, right? Yes, there are many other things, but IgA is one of the component, right? In your genitourinary tract, okay, in your digestive tract. So in all these secretions you have IgA, okay, and it's produced in very high amount, so 5 to 15 gram of immunoglobulin. So you can just imagine, it is such a small thing, you cannot even see it through your naked eye, but it is 5 to 15 grams of IgA released in secretions, right? And IgA, if it is present in the serum, it is mainly in the monomeric form, okay? But in the secretions, it is usually in a dimeric form, okay? And you have already seen how the dimer look like. Okay, so if we talk about IgA, then the first thing that should come into your mind is mucosal immunity, okay? And again, a question, a very important MCQ based question is that which of the following immunoglobulins are present in the external secretions, right? So you can just imagine that IgA is uh, protecting when some pathogen is trying to enter, right? So it could be through mouth, it could be through eyes, it could be through nose, it could be through the urinary tract, it could be through your genetic areas, it could be in your digestive tract, right? So in all these regions, you will have IgA to protect you from pathogens, okay? Now, uh, as I said, in serum, it is usually in a monomeric form. Okay, but in secretions, it is usually found as a dimer or tetramer. Okay, so tetramer is rare, but usually a dimer. Okay, dimer means this two molecules of IgA. Okay, plus a J chain, right? So if we talk about dimer, it consists of two molecules of IgA. It also have a J chain, right? And it has a secretory component. Okay. So students, let me show you one picture. We have already seen that, but I'll just show you again. तो हम घूम फिर के इसी इमेज पे आ जाते हैं, राइट? So you can see here when I said IgA, so it consists of two molecules of IgA. So this is one molecule and this is the second molecule, right? So two molecules of this, and then we have this J chain, right? And the secretory component, so this red one, right? So this is the secretory component. So when we talk of IgA dimer, right? So suppose, students, I am again emphasizing, suppose you get a question to describe the structure of, suppose, a dimer of IgA, right? So in that case, you have to tell that IgA is composed of one, two molecules of IgA, two, the J chain, three, the secretory component. Okay, clear?
Sir, can you repeat?
Can you repeat?
Yes, so I am saying if we have to talk about the structure of IgM, no, if we have to talk about IgA dimer, okay, so if we are talking of IgA dimer, then it consists of two molecules of IgA, okay, it contains the J chain, okay, and it contains the secretory component, okay, this red one. So I will tell you maybe in the next class from where this secretory component is coming, okay, what is the importance of this secretory component, okay. So I was just saying that if suppose you get a question to describe the structure of a dimer of IgA, then you have to tell all these 3 points, that it constitutes of two IgA monomers, a J chain, and the secretory component, which you can see in this picture as a red line here. Okay. Okay, so as I said, so it consists of two molecules of IgA, a J chain, and a third one is your secretory component. Okay. Now the secretory component of IgA protects IgA from being degraded by proteolytic enzymes, right? So students, you can just imagine this IgA dimer has to survive in the harsh gastrointestinal tract conditions, right? So if suppose this dimer reaches in that gastrointestinal tract, right? So the environment is harsh, right? There are a lot of degradative enzymes, there are a lot of proteolytic enzymes. They will cleave this dimer, right? So to protect this bond, right, this secretory component helps. Okay, so this secretory component, it covers the regions of IgA dimer, okay, and it does not allow these proteolytic enzymes to cleave it. Okay, clear? So secretory component protects IgA from being degraded by proteolytic enzymes, and it helps in surviving it in the harsh gastrointestinal tract environment and provide protection against microbes that multiply in body secretions, right? And IgA is a poor activator of complement system and it opsonizes weakly. So IgA का काम is not to activate the complement system or help in phagocytosis. IgA का काम ही different है. It has to work in different areas. Okay, clear? So you can just imagine complement system is not there in external secretions, right? So it doesn't need to be a good activator of the complement system. Complement system proteins कहाँ है? वो circulation में है, system serum में है, blood में है, right? So where is this dimer? It is there in the external secretions, right? Or in the harsh um, uh, gastrointestinal tract. Clear? Okay, so in the next class, we will talk about how this IgA dimer forms and from where does this secretory component comes. Okay, and another very interesting question, students. This molecule is formed in the serum, in the blood, right? Then how it moves into the external secretions, how it reaches the milk or how it reaches the mucus, right? So that we will discuss in the next class. Okay, so any questions? Okay, so because the time is up, so I'll just stop here and we have still a minute to go. So if you have any questions, you can ask. So today we have talked about IgG, we have discussed about IgM, we have discussed the pentamer of IgM, the importance of the IgM pentamer, right? How IgM pentamer is formed, what is the clinical significance of IgM, right? It helps in identifying whether it's a recent infection or not, okay? So that helps in diagnostic. And then we have talked about IgA, where IgA is present, what is the importance of IgA, okay, and the structure of IgA dimer, okay. So in today's class, we have covered so many aspects of these different types of antibodies. So we have covered IgG, we have covered IgM and IgA. In the next class, again, we will start with IgA and then we are left with IgE and IgD. Okay, the good thing about IgD is that we actually don't know much about IgD, so there is nothing much to read related to IgD. But yes, IgE has a very important role in hypersensitivity reactions, so that we can discuss. Okay, again we will not discuss in much detail because later we have to actually study hypersensitivity, so we can study those things in detail when we read about hypersensitivity. Okay, so I'll just stop here and see you in the next week. Okay, okay
Thank you, sir.
students, okay,
Thank you, sir.
okay.